I want to talk about mining different databases that are important for exposome for a particular subtopic, the blood exposome. Before going into the topic, I want to thank funding agencies for expanding funding and allocating more than $300 million from NIH and European Commission on exposomics and metabolomics. And hopefully we will add an extra zero to that 300 number pretty soon. These are the discussion topics for this talk, that how we can prioritize chemical in exposome field, how do we develop a chemical to publication mapping approach for the blood exposome database, and how metabolomics can be used to expand the blood exposome database, and what are the important chemical databases for exposome. Question one, how do we prioritize chemical in the exposome research? Most exposures are chemicals, whether they're coming from different routes, dye, drugs, altered metabolism, air, water pollution, they all chemicals. In fact, if you look at one of the key agency who evaluate exposures, IARC monograph, 82% are individual chemicals out of total agents, 1,079. And there are mechanisms in place in government and non-governmental agencies to identify, monitor, and regulate exposure to a specific chemical. So when IARC monograph does the classification on that a particular agent is carcinogenic or not, and they use a classification scheme based on availability of evidence in primary literature and other relevant toxicological databases. So in this classification scheme, you can see that group 1 has 120 agents that there is a sufficient evidence in literature and other databases that these 120 agents can cause cancer. Now, I wonder if similar classification should be developed for chemicals for other diseases, such as diabetes, Alzheimer's disease, or cardiovascular diseases. And so IARC monograph is a great example that how chemical agent evaluation takes place for a non-communicable disease. And the way they work is agents are nominated by public, and it goes through expert judgment, public health importance, and advisory group. Advisory group get together, and then they recommend that in follow-up 4 or 5 meetings, these are the agents that will be evaluated. For example, a meeting 128 will be held, and acrolein and other chemicals will be evaluated. And it takes 2-3 days to go through different evidences and literature, and then final report comes out after almost a year. So they have done almost 125 meetings. And the major requirement, they always look at what is the availability of evidence. So I had a privilege to work with IARC monograph team to analyze availability of evidence in literature data for pesticides. So in this picture, we see organophosphorus on the top part and organophosphorus at the bottom. Size of the node represent how many papers are on pesticide and cancer, and border of the dots represent how many are actually from cancer epidemiology. Analysis included almost 1,000 chemicals, but here we look at 2 major chemical classes. And based on this analysis, IARC monograph organized 2 meetings, 112 and 113, on selected pesticide agents using recommended by this analysis. So now this analysis included text mining and use of chemoinformatics to mine entire PubMed and several other databases to put in a way that help the advisory board that what chemicals can be recommended for next evaluation panel. So inspired by this work, that can we prioritize chemical for some other biological context? And when we think about that, how many papers we need to go after, the volume is really large. We all know that there are 1.1 million paper that can be considered original research articles. So they're not review, not letters or anything. They're original research article. Almost 18 million total original research article. And we need to connect chemicals to publications in those 18 million papers. And then we will be able to actually prioritize chemicals for different contexts, like how many compounds in blood, how many compounds ever reported for tissues such as liver, or dry blood spots, or for particular disease, organisms, and so on. So that mapping need to be done, and there are computational approaches doing it. I try to put them together into a fully automated text mining and cheminformatic engine that allows connecting chemicals to publications, so we can do this contextualization. So this goes to then how do I use that chemical to publication mapping to develop the blood exposome database. So when we talk about chemicals, origin location is important. So if you look at these 4 triangles, outer one is all known chemicals, about 100 million of them. So in a red triangle, chemicals in exposure, we don't know the estimate for that, how many chemicals we actually have in our exposures. And the blood exposome database, it's a yellow triangle that some of these chemicals from exposure gets into bloodstream. And so I try to estimate that. And then you see the tiny red triangle that out of them, how many were actually evaluated that they were hazardous or beneficial to human health. And number is about 2,000. So what chemical constitute these triangles? And I will highlight the blood exposome one. So if you think about how many papers do I need to look at, and it's about 70,000 papers. Something was measured in blood every year. Together, 1.4 million publications to go after. How people report chemical names, that you can have abstract chemical names are individually mentioned and full text either in a table, figure, in paragraph, or supplementary table. So we need to mine chemical names or look for chemical names in all these different portions of a publication. Of course, there's the issue that blood is such a generic term that people can use like blood pressure or blood transfusion. These terms exist, and these are false positive hits. We can remove them using a phrase pattern exclusion list that if those phrases occurs in a abstract or paper, we can remove it. And we can shrink the list from 1.4 million paper to 1.1. So next question is, from where we get the chemical names. Here PubChem comes as a really handy database that we can make a subset of entire PubChem for relevant databases, and that will give us about 15 million synonyms to search against. For a typical normal well-known metabolite or chemical, you will have about 20 to 30 synonyms. And it's really important to know about all the synonyms because if you claim that this is a novel metabolite and use a totally different name, and then other people will complain that, oh, I know this chemical, but you use a different name. So just naming can cause confusion that this is an old metabolite or chemical, or are we talking about the same thing? I'm not going to do it manually, of course, because reading 1.1 million paper, it's just laborious task, and that green dot shows that how many people HMDB effort has been looking at, so it's almost 2,000 paper in 10 years. So there is no way we can tackle this by manually going through all the paper on chemicals in blood. So we're going to do it computationally, and there are 2 ways to do it. Crowdsourcing, that we have a central hub where everybody deposits the chemical to publication mapping, and then we can do automated mining, that we take all the publicly available literature data and link them to chemical synonyms. So now this is fully automated. So there are 3 processes here. The bottom one is chemical to synonyms, a simple text search. Middle one is a PubMed central supplementary table and their connection to PubChem. And top one is PubChem provides a consolidated list. Whoever submitting data to PubChem, they will link it to PubMed. So we can get 3 lists, and then when combined together, and then filter by PubMed abstract, that will give you a master list of that how many compounds we will have in, how many compounds were ever reported for blood specimens. So to search chemical names in such a large number of publications, so we can't do a normal text string search in R or Python, but I like to use a database approach here, and Elasticsearch is a wonderful database here, and that we can index millions of papers in it and search all 15 million abstracts. And if we do that, we get 39,000 unique chemicals linked to 851,000 publications using chemical name search. Crowdsourcing by PubChem. So PubChem provides this file where its chemical and publication are linked on their website. From FTP we can download, and it gives an idea about that who is submitting data, that there are individual depositors, and most of them coming from medical subject headings. Then we have publisher and bioassays who submit data to PubChem with publication. It's important that everybody submit whatever you report, analyze in blood, please submit to PubChem. This is a nice example, N-lactolide leucine. It's a blood compound, but it wasn't mentioned in the abstract. And so if you do a Google search or PubMed search, you will not find this paper reporting that this compound was actually detected in blood. It was only mentioned in full text. And luckily, the institute actually submitted these 5 chemicals in PubChem, and I was able to find out that, okay, this is a nice example that why, if you measure blood chemicals, you want to submit your PubChem to PubMed mapping. And that PubChem will list your lab or organization in one of those 722 PubChem sources. It is so easy to submit data to PubChem. It's only 4-column table. I can help you if you need help in submitting those lists to PubChem. And I'm sure it will increase citation rate for your paper. If you report a new compound and you want that people cite it, this is a good way to do it. So third approach is PubMed supplementary data. So we have 11,500 papers, open access, or 7,700 in PMC, where we can download supplementary data using an automated API. So in PMC, you can submit a paper, but they have a copyright that you cannot download many papers. So you can read it online, but not all PMC papers are downloadable. So out of those 7,700 papers, we can parse out 4,000 PubChem CIDs. And these are only metabolomics paper. So these are supplementary tables in metabolomics, blood metabolomics, and there are 4,000 compounds in them. Gives an idea that how many compounds metabolomics field have identified and reported in public domain. So if we put together these chemical lists, PubChem to PubMed mapping provides the highest coverage for blood-related compound, and compared to HMDB primary evidence, so linking of chemical to a publication. So HMDB has only 1,000 chemical, where primary evidence for reporting a chemical was there. And metabolomics provide additional 1,250 compounds that were not covered by PubChem to PubMed or HMDB. And if you look at altogether, this is about 42,000 unique two-dimensional chemical structure that were ever reported for blood-related publications. And if you look at properties, that the distribution of their mass, lipophilicity, it's 1 point something, so it's close to semipolar, and the majority of them are below 1,000 Dalton, and almost 3,500 have at least 5 publications or more that they were reported for blood. So we published a paper in EHP last year, and it's a nice database, web accessible, downloadable, and you can query by chemical, publication, so on. Potential application for this particular database could be prioritization. We can use it for targeted asset development, compound identification, mass spectral library development that is happening at WCMC. Also biological interpretation that chemical are linked to publications. And if you query some chemical, want to know about that, if any other context the chemical was detected, you can query the database to get it. So that was introduction to a new database, Blood Exposome, that will automatically mine new publications and databases and compile these lists for blood-related. And we will roll out, in my next job, I will roll out a fully automated system for other biological contexts, and those lists will be available on GitHub. That every week, those lists will be uploaded to GitHub. So how metabolomics can expand blood exposome database? So measurement is key, and there are two main factors in measuring chemicals, chemical diversity. So this is Steve Rappaport's paper. I actually made that network plot with Steve, that we have huge chemical diversity in blood, and we have concentration ranges. So what it warrants that we can't use one type of analytical method. We need to combine different approaches to cover or expand the chemical list for blood measurement. And of course, we need to use targeted and untargeted both approaches. And the fundamental difference is that in targeted, we have an analyzer that just filters the signal that we record. In untargeted, we don't have that filtering. We record as much signal as possible. And there has been tremendous progress in metabolomics for blood that in last 11 years, there has been 12-time increase in number of identified compounds. So in 2008, 150 compounds in a typical report by a metabolome for blood in all literature. This is all public domain. And now if we see, we have reports with 1,700 identified compounds in 2019. And all those new annotations are coming up in this Venn diagram that the metabolomics will be a key technology to expand the blood exposome database, that it will add new and new chemicals. That was by a company, and in public domain at UC Davis, we were also combining multiple technologies, so reverse phase, helix, targeted, GCN, different combination of analytical chemistry to measure. So in this paper, we had 900 uniquely identified compounds. Today, their number is close to 1,300 uniquely identified compound for blood specimen. And WCMC is a service center, so anybody can send sample and get the data for these 1,200 to 1,300 compound for a typical blood sample. And we all heavily debate the peak annotation is an issue in metabolomics and confidence scoring, and I have a ranking that the good annotations where we have MZRT and MSMS, and OK and bad where we're missing one of these two. And there are ugly annotations where we only use M over Gs, because it's important, because when we're trying to propose that we identify a new compound and that IARC monograph or any other agency should utilize the data, then annotations should have the highest possible quality. We can't rely on MZ annotations for recommending chemical exposure for evaluations. Now, large-scale untargeted metabolomics is feasible for blood specimen if we use machine learning methods to correct batch effect and signal drifts. So in this paper, we highlighted that if simple random forest machine learning method can get rid of most of the technical variance and the batch effect we introduce by sample preparation or instrument variability. And we call it SURF, systematic error removal using random forest. And we have tested that on 1,000 sample, 5,000 sample. It works beautifully that most of the analytical and technical variance can be removed by machine learning based batch correction or signal correction methods. Next point for blood metabolomics is we don't want to handle like 50 or 100 samples. We need to think about the handling samples in the range of million or 100,000. And if we want to go in that direction, we need to start thinking upfront that how are we going to put the sample, the collected data in databases. Such an approach is working greatly at UC Davis for GCMS. The database has almost 200,000 samples collected in the last 15 years, and you can actually query a sample collected 15 years ago and today. And that gives an enormous power to compare that if a biomarker is specific, how many times we detected this compound in any other context. Now, this database approach we need to have for LCMS-based metabolomics, and that way we can look at data, untargeted data. For targeted, it's easy. For untargeted data, then we can look at that how metabolome changes over time and how that can be associated with outcome data. So the last point of my talk is, what are relevant chemical databases for exposome? The most important is PubChem. And the PubChem database has evolved tremendously in the past decade. So over 100 million compounds are there. And now PubChem is collecting information about toxicology, metabolism, pathway, and becoming a great knowledge base. For example, benzene has 96 pages of rich information. And many toxicological databases already has been incorporated in PubChem. But not all relevant databases for exposome are in PubChem. So I try to group these different databases by international agencies, so OECD, Labor Organization, WHO, United Nations. They have committees, they have panels, they make these chemical lists, but many times that doesn't go in PubChem. But nonetheless, there has been great effort by government and non-government agencies to make these chemical lists, and these lists are all relevant for PubChem, for exposome research. There are about 115 relevant chemical databases in these categories. So then we have government registries, pharmacology and clinical chemistry, which include all the drugs and FDA drugs and clinical chemistry measurement like Mayo Clinic Laboratory Catalog, and risk assessment by EPA, NTP, IARC monograph, NSAS, and so on. And here we also have pathway, metabolite, and ontologies. For example, HMDB, Reactome, SABI, and so on. So there are more databases will be coming in these categories. And the goal, I think the goal will be that how we annotate chemicals from one database to other one. And this way, a database become an implementation point. So implementation endpoint for blood exposome. If you look at the prevention spectrum, that we have primary prevention, secondary, and tertiary prevention. So on the primary prevention side, there are substance registries, a risk assessment database, and biomonitoring. So it's a CDC enhance or IARC monograph, or we have substance registry, EPA or FDA. Now think about that we detect a chemical using untargeted metabolomics for blood, and that we think that it's related to cancer, then we can recommend that to IARC monograph that please look at our publications and think about this chemical in the next evaluation panel. In middle diagnosis and screening, we have electronic health record and clinical chemistry, and they have a certain, they have a list of chemicals that they measure using targeted assays. And blood metabolomics or exposome research can recommend new chemicals that can be included in those blood chemistry tests. And for developing chemical monitor and analysis of dry blood spots in targeted way, the research can recommend new chemical candidates. On tertiary side, I like Bob's example yesterday that how we need to look at from a subject, as a patient who got the disease, and how we can measure new chemicals that can affect the outcome. So those chemicals, once we discover in our primary, once we report in primary literature data, we can recommend them to blood chemistry panel that they must be included in those panels. So just an example of primary prevention implementation endpoint. This is a PrEP 65 list by California, and it has 777 chemicals. It will be question in front of us that how exposome community can recommend new chemicals that should be included in PrEP 65 or any other hazard assessment agencies. And implementation second, the blood chemistry or EHR data that Mayo Clinic has 900 chemical listed. And if a physician or doctor recommend any of these cases, assays, it get connected to electronic health record. So what will be the new chemical that should be included in this clinical chemistry panel? So this is where blood exposome or blood metabolomics can recommend new chemicals, either on the primary prevention side for risk assessment, or also secondary tertiary prevention, that what new chemicals can be monitored in the patients. So in conclusion, prioritization and expert evaluation of chemicals for different diseases and biological contexts is an important process in exposome research. We've got to think about it upfront, that how we will utilize all the primary literature and knowledge we will generate using our exposome research. What is the missing dot? And how we can think about a classification scheme like IARC monograph for other diseases and outcomes that we are interested in. And computational text mining can improve prioritization process by linking chemicals to publications. Blood exposome database has been delivered, and we can automatically update it using a workflow of text mining and cheminformatics. And we gotta use machine learning to correct signals in untargeted metabolomics on epidemiological scale. And exposome databases by different expert panels or even individual laboratory or organization should be consolidated in a central hub such as PubChem. And PubChem provides a nice mechanism to store that. So with that, I would like to thank all my collaboration at UC Davis, my friends at IARC Monograph, Rima at Duke, Steve Rappaport at UC Berkeley, Ian Lipkin at Columbia University. And thanks to funding agencies for different grants that supported me in the past many years. And I'm looking for postdocs. If you're interested in any of these topics, reach me out at GitHub. Don't send me email. GitHub is a good way.